Okay. So today I'm going to continue talking about event-related potentials. You're definitely have some readings going on right now with some various event-related potential studies. We have one or two review articles related to event potentials. And those different articles that you're reading are going to highlight some of the different event-related potentials that I'll be talking about in the lecture. So in addition to talking about event-related potentials, I am going to offer an extra credit opportunity for Friday. So if you notice, if you look at the syllabus, the lab syllabus, there are no more scheduled labs for the remainder of the course, but it says things like meetings or data collection, which means that the times you normally use for lab are a good time for you guys to do stuff related to group project 2. The extra credit opportunity that you'll have will be related to the coding that you will have to do for group project 2. And you do not have to be done by any means. But basically, for everyone who shows up in your group, I will give a extra credit, up to 3 points of extra credit. And I'll give varying levels of extra credit depending on the amount of coding you already have done. But really more the questions that you have, if you are, problems you're trying to solve, and the ability to to recognize what things you might need help or you're getting stuck on, errors you're having. So like our ability to be productive as far as getting some of the bugs out of your code. So up to 3 points extra credit. And that will be if you can make an appointment with me Friday on Zoom, this coming Friday, to talk about your code for your experiment. Any questions or comments about that? Okay. So additionally then, just talking about event-related potentials for a few minutes today. And this comes out of talking about EEG, frequency analysis studies. So frequency analysis studies, when we're using Fourier analysis to decompose the electrical activity on the scalp, the EEG, into the different components, delta, theta, alpha, beta, gamma. And then amplitude is oftentimes the main measure, or maybe power, which is very similar to amplitude, is the measure in a lot of frequency analysis studies, comparing the alpha amplitude when the participant is doing one thing compared to alpha amplitude when the participant is doing something different. And I mentioned there's a couple important differences for event-related potentials. I believe you guys actually answered some questions about that for the in-class assignment. Last time, event-related potentials, one of the primary differences in event-related potentials is that there is no frequency analysis in event-related potentials. So this thing right here, this is an ERP. This is just like kind of the average raw electrical activity on electrode occurring in response to one of these two types of things, but no Fourier analysis. This is the raw, unprocessed electrical signal as recorded on the scalp, though it's been averaged over many trials. So these different waves are the average response over many different trials of seeing these types of objects right here. So one thing that makes ERPs different than frequency analysis studies, ERPs, there's no Fourier analysis. There is no different frequencies that you're looking at. The other difference in ERP studies compared to frequency analysis studies, usually in ERP studies, you're comparing amplitudes between at least two different conditions, and you're doing so in a very small range of times. When you're doing frequency analysis studies, Fourier analysis, kind of the technique of decomposing into the sinusoidal components requires that you are averaging the response over kind of a long period of time, well, kind of long, like 250 milliseconds or at least 100 millisecond range when you're doing Fourier analysis, frequency analysis. You're always looking at amplitudes over a relatively big window. With event-related potentials, when you're not decomposing into those different sinusoidal components, the window can be much smaller. And so these have arguably higher temporal precision, event-related potential analysis, than frequency analysis, because you can look at very specific time points like the N170 ERP. The N is a negativity that occurs 170 milliseconds after the response to an object, and the amplitude of this event-related potential is bigger for faces, which is this right here, compared to cars. Negative is down, positive is up. The N170 is the difference between these two waves that occurs about 170 milliseconds after the onset of these objects. So that's just some review. So today we're going to be talking more about event-related potentials. And I mentioned last time, we're going to be talking about a lot of them. You'll read about a good deal. You won't read about all of these, but you'll definitely read about a good portion of these. I mentioned there's kind of two main categories of event-related potentials. Those which are tightly time-locked to some stimulus. That's the main type. A lot of research uses those, but there is kind of some exceptions to the rule. There are some event-related potential studies which are less tightly time-locked. When you're looking at amplitudes, electrical amplitudes, over a longer period of time rather than a really small period of time. So in these studies, usually you're not performing a Fourier analysis. You're still just looking at averaged EEG amplitude over a span of time. The main difference here, though, is that you're looking at longer spans of time, so it's not as tightly time-locked. So we won't get to these today. Today we'll talk about brainstem potentials, sensory RPs, and the mismatch negativity. These are just three different wave components that are studied in event-related potential studies. We'll talk about these today. Okay, so when we're talking about the brainstem potential is measured with electrodes, of course, on top of the scalp. And I've mentioned that before, the EEG and event-related potential studies don't really have that good of spatial precision. That is, these electrical signals that we're recording from the scalp, for a whole bunch of reasons, it's not exactly clear where they're coming from, or that is, you can't use the signals themselves to kind of figure out where those signals are coming from in the brain. However, there is this thing right here called the brainstem potential. It's an event-related potential response, which is, so it's electrical activity measured on the scalp. And there's pretty high confidence that this event-related potential is coming from roughly the brainstem. And so when I'm saying brainstem, actually that picture's not that good. This picture's a little better. So the brainstem is thought to be, I'll go back to that slide in a second. The brainstem is thought to be one of the very first places where sound information from out in your ears enters the brain. So right here, here's a whole bunch of different facial nerves, or cranial nerves as they're called, which their job is to kind of take in sensory information into your brain, and then they also can output motor information out to the effectors like your mouth, the different muscles that you have in your face. So they're kind of like input and output wires. And I'll be talking for these event-related potential studies, brainstem potential studies, we'll kind of be studying the input wires. Those input wires, like the acoustic nerve, which presumably takes in information from the sound from your ears, and then that information enters approximately in the brainstem. So I'm not going to go into like the microanatomy of the circuits down in here. Basically, for our purposes, there's just several different brainstem or subcortical structures. Technically, this is like the brainstem and the midbrain, and there's kind of connections both in the brainstem and the midbrain. So really, these are all, it's called the brainstem potential, but the brainstem potential probably emerges both from what is technically the brainstem, but also from some signals which are like the midbrain, in particular, the superior and inferior colliculi, which are located in the midbrain. So the brainstem potential is really coming from the brainstem and the midbrain, but it's oftentimes just called the brainstem potential. And so brainstem potentials, they are increases or bit fluctuations in the electrical activity that occur in response in particular to sound. So sounds appear to be the sensory modality, which, all signals go to the brainstem at some point, but the very first place that sounds go to is the brainstem, and there's a lot of processing apparently that occurs in these subcortical structures in sound, and the processing appears to be more of it than, for instance, the visual systems. The visual system goes to these subcortical structures as well, but it appears that sounds, there's more processing, there's more things occurring with the representations of the sound in these subcortical structures than in the representation of things that you're seeing. There's information in both visual and auditory in these subcortical structures, but there seems to be more stuff going on with the auditory processing. So I guess I do have a picture of a little bit of the microanatomy. So these are kind of, I stole these from my cognitive neuroscience class right here. Sound would enter the ear, and then the auditory nerve right there. And then it kind of makes its way eventually from, starting, these are all subcortical structures. So these are parts that are underneath the cortex. Cortex is up top. The subcortical areas are all beneath the cortex. And these are supposedly kind of some of the different, I'm not going to go make you remember all of these things, but this right here is technically part of the thalamus. The inferior colliculus is part of the midbrain. The superior olivary nucleus is supposedly part of the brainstem. And all of these are potential sources of the brainstem potential. So it's not 100% sure which of these, or if there's one particular part of this which is underlying the brainstem potential responses. It probably is all of them. The reason why researchers, because as I mentioned, EEG studies, ERP studies, it's hard, you can't really localize the specific pattern of electrical activity to any specific brain region. So you might be wondering, well, if you can't localize the electrical activity to a specific brain region using the EEG signals, how do I know that these brainstem potentials that I'm going to be talking about, they actually come from these subcortical brainstem midbrain areas? And so there's not direct evidence from the EEG studies per se, but if you look at other studies on animals, it makes these conclusions about the brainstem potential, its origins being the subcortical areas, much more plausible. And it's these intracranial studies in animals. They provide strong evidence for this auditory pathway that includes the subcortical structures right here. And this is like the really crucial piece of evidence. In animals, when you present a sound and then you measure from these areas. So in animals, you can actually stick electrodes directly into the brain. You can't do that in humans. In animals, when you do that, you can measure the latency of these electrical responses in the animal. So you present a sound to an animal, and then eventually that sound becomes represented right here. But that sound, is the information from out in your ear, does not reach the auditory pathway instantaneously. There's a little bit of a latency, 2 to 5 milliseconds. It might actually be as big as 2 to 10 milliseconds of a latency. The information hits your ear, and then it's being processed apparently in the brainstem between 2 to 5, maybe 2 to 10 milliseconds later. So as I said, this is what happens in animal studies. They measure directly in these subcortical structures, and they see that there's a latency of about 2 to 5, maybe 2 to 10 milliseconds. When we're doing the brainstem potentials in humans, the inference that these brainstem potentials are coming from these subcortical structures has to do with the fact that what we are doing is we are focusing in on the electrical activity, the fluctuations, the amplitude in the signal that occurs with latencies, very, very short latencies. This is incredibly short amount of time. So 5 milliseconds is 1/200th of a second. So that is that's a sliver of a second right here. And so these are the same latency. So these, in animal studies, it suggests that sounds is reaching this part of the brain 2 to 5, maybe 2 to 10 milliseconds. When we're measuring brainstem potentials in ERP studies, we're looking at those same latencies. We're looking at the electrical activity between 2 and 10 milliseconds, and we're making the inference. Well, if it's occurring with the same latency as it does in animals, these responses are probably coming from the brainstem. So that's how one of the ways you can make inferences about where a EEG signal is coming from, in this case, these brainstem potentials. You can do it based on inferences, what happens in animals, and then you can kind of focus when in time you focus your amplitude comparison. And that helps make the inference that we're probably studying the brainstem here. So here are this is like kind of the main, I've kind of done a little bit of research and preparation for this, and these are kind of like the main stimuli that have been used in studies of the brainstem potential. So clicks are oftentimes stimuli that are used in clinical applications. So the brainstem response, which you can see kind of a idealized one right here. So this is like a response. This is taken from an open access article. This is basically the response of a person's auditory pathway, these subcortical structures, in response to one of these clicks. So what these clicks are is just a whole lot of clicks that occur. They're really short, so short that I can't even do an impersonation. Very short clicks that occur over the span of like 500 milliseconds or maybe a second. I think an article I read, it was like 40 clicks per second. So it's a lot of clicks happening per second. And that, it's really quick stuff. But what they're doing is they zone in on one of those clicks, and they're looking at what is the subcortical, the auditory pathway, the subcortical, the brainstem's response to one of those clicks, kind of in this sequence of random clicks that occur that the person hears. And a common clinical measure that uses the brainstem potential is the latency of this peak right here. So you look, once again, you look at the brain's response to one of these click sounds, and that's what this is. And then a common measure is the latency of this peak. So usually when there's no sounds, the brainstem electrical potential will be around 0. It'll be flatline. There's nothing going on there. But then when a sound occurs, even something like a click, there'll be an amplitude fluctuation, an amplitude increase. And that's what's being shown right here. There's a large change in the amplitude from like the 0 right here to this. And then typically the way the latency is measured is by the peak is right here. And then you look at how much time occurs from the onset of the stimulus, the click, which would occur, some point in time, like right here. And then there's a little bit of a delay. Remember I said animal studies, it's like between 2 to 5 milliseconds, maybe 2 to 10 milliseconds. There's some delay until the response to that stimulus, there's a peak right there. And so a common dependent variable is the amount of delay between the onset of a click and then the onset of the peak electrical response that supposedly is coming from the brainstem. This is the brainstem response. So clinically, this has been used a lot to diagnose various types of neurological conditions. Generally, people that have neurological conditions will have delayed latencies. So a person who does not have a neurological impairment, their latencies will be relatively quick. So they'll hear a click, and then 2 or maybe 3 milliseconds later, they'll have a representation. They'll have this peak in their brainstem response. But individuals with different types of impairments, usually hearing impairments or neurological impairments, their latencies will be relatively longer. It'll be more like over here. So that's a common diagnostic thing for different neurological conditions, the latency of this. And I'll show you some specific information about that in a second. I've actually read a good deal about this research right here. Oh, is there a question? Yes. I don't actually know about these, whether it's usually, I think these are usually considered positive amplitudes. But that's funny. In these, I have read a lot of these studies. They don't really call them positivities and negativities as much. And I don't know the reason for that. But we'll see some more pictures and maybe we'll be able to tell. Yeah. Okay, so the reason here is a positivity. So here's another one. I'm zooming ahead. The first fluctuation is right here. And then there's another fluctuation right here, which is a negativity. So like this wave right here, they call it in this study, the V wave is a positivity because it's on the positive amplitude side. But then there's another response right here, which is a negativity. So yeah, I don't completely understand why they don't have the same naming scheme for these brainstem potentials as they do for ones like the N170. They have a completely different naming scheme, and it's probably just because different researchers are doing them, and so they come up with their own different naming schemes. Good question. And I'm jumping ahead a little bit, so I'll come back to this one and definitely explain that one better here in a second. Yeah, so usually I think the first response is a positivity, but then there will be some like reverberations later that can be negative response. But usually this is like the onset response. It's 0 when you're not hearing anything, then you hear a response. And as far as I understand, usually that first response is a positivity, but it's also oftentimes followed by deflections to the negative side, which are kind of shown right here. Big positivity, and then usually it will go negativity after that. Okay, so yeah, the other speech, these are the ones I've read the most about so these are usually used, the clicks, stimulus are usually used in like clinical research for whatever reason. There's a lot of basic research where they'll just use speech. So they'll present speech in either like running speech like in sentences, or actually what I've read usually is they'll just repeat the same speech sound over and over and over again. So instead of repeating clicks over and over again, it'll be like the speech sound da, 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 which doesn't sound particularly The stimuli are not particularly fun to listen to, but you repeat those DA sounds over, and then you can look at what does the brainstem, how does it appear to be representing speech sound information? And at a basic level, there's some similarities. So once again, this was the onset response. This is like the increase in amplitude that occurs in response to a click sound. And there's a similar onset response for a speech sound. So, and then I say something. For a while, when I went during that little silence just then, your brainstem potential electrical response should have basically been flat. There's going to be a little bit of noise, but it's basically going to hover around 0. But then as soon as I say syllables, the amplitude is going to get bigger. And this is the onset response to speech. And similar things, they find that for different clinical groups, and I'll show you some evidence here in a second, they find that the responses tend to be delayed in individuals that have neurological impairments. So there's always a little bit of a latency, a little bit of a delay from the time the sound actually occurs to the time it's represented in the brainstem response. In general, people with higher cognitive ability appear to have shorter latencies compared to people oftentimes who will have neurological impairments. The latencies, whether it's a click stimulus or a running speech stimulus, the latencies will be delayed in neurologically impaired people, typically. So here's some evidence supporting that. So once again, yeah, here they use different naming schemes for these as they do for like the N170. They use different naming schemes. Sometimes they call these waves 1, 2, 3, 4, 5. Here they call this wave V and they call this one A. So different naming schemes. But this is showing in a little bit more detail. So these are kind of like idealized responses, but this is an actual response from a study. And so up here, this is just like, this is right when the stimulus occurs or is presented. That's the 0 time point. And you can see we're talking about really small time scale here. The entire graph is zooming in on 15 milliseconds. So that's, a 50th, less than a 50th of a second. So incredibly fine temporal precision of this response. And in general, there'll be kind of, just little noisy fluctuations that occur for a while, but then at some point, there'll be an amplitude where there's a big amplitude extension in both directions. And that appears to be, they kind of arbitrarily mark these sometimes. Like, it's hard to know, is this the brainstem response right here? Or is this the start of the brainstem response right here? So there's a little bit of subjectivity to where you decide the wave fluctuation occurs. But here you notice that it's always going to be somewhere between 0 and 10 milliseconds. The general assumption is that the electrical responses that are occurring in the EEG, up to 10 milliseconds after a sound is presented, the general assumption is that those are coming from this subcortical structures. So here, in this study, they believed that this V, that's what they consider the onset response. In this A as well, they considered kind of like the deflection of this. So this is a positivity here and then a negative. So you consider this like a deflection. You have an amplitude increase in the positive side and then kind of a corresponding negativity down to the negative side, corresponding amplitude fluctuation to the negative side. And then in this study, the two different participant groups that they were comparing, TD stands for typically developing children, and ASD, autism spectrum disorder. And so the ASD, the individuals, the children with autism were the light gray line right here, whereas the typical controls, they're the dark gray line here. And so you can see at the beginning, they're kind of matched up here, but the really crucial comparisons that they care about, at least clinically, are the variations in latency that occur in these peaks right here. And you can see this one right here is a little bit small, but there is the peak latency of the typically developing children is a little bit before here for the V wave. It's a little bit earlier than the children with ASD. If you look down at the deflection, though, there's a big difference by this point in processing, where the typically developing children, the latency of their peak response, which is right here, is clearly earlier than the peak latency of the children with autism spectrum disorder. So as I mentioned, this is using speech stimuli, I believe. Yeah, the syllable da. So this is the brain's response to the syllable da, said over and over and over again. the brainstem response of particularly the A wave, which is the negative deflection, that the latency was earlier for controls than it was for the neurologically impaired or the autism spectrum individuals. So it's a typical way of using the brainstem potential as a clinical biomarker. So here's a little bit more like studies about, this is how the brainstem potential response can be used for studying other groups. For instance, musicians, they're not I was just talking about, neurological impairment. Musicians in some ways are considered to be kind of the opposite. Their brains in some ways are process sounds in particular faster than typical controls. So neurologically impaired people, oftentimes their brainstem response is late, but musicians compared to controls, but musicians on the other hand, their brainstem response tend to be earlier. So there's kind of like this continuum of, neurologically very strong, the onset responses, the latencies are very small, and neurologically impaired, the onset responses tend to be a little bit later. And this is a study where they're kind of looking at the way that onset latency, which is this is what these are right here. So you can ignore the stimulus to response correlation stuff. I mean, that's a common thing, but it's a little bit more complicated than I want to talk about. The latency correlations are right here. So here, the onset latency, that's that V wave that I just showed you. And then the transition peak, that A wave right there. Looking at correlations between those latencies and performance on these are speech and noise tasks. And then so they find that the longer the peak, it's like negatively scored. So the longer the peak, the worse your performance, because these are signal to noise ratios. And so signal to noise ratios is like a negatively scored dependent variable. You're doing for signal to noise ratio, your ability to perform the task is bad if the signal to noise ratio is big. For a speech and noise task, you're trying to hear speech sounds, and there'll be like a bunch of babbling noise. Like if you're at a party and you're trying to listen to someone who's talking, but there's a bunch of other people that's talking, you're trying to focus in on a speech signal while there's a bunch of competing noise. And it's a little bit hard for the brain, particularly when you get older, you get much worse at hearing speech and noise. And so they do this type of task in the laboratory where you're trying to hear a person talking, but there'll be a bunch of background noise. And here, the signal to noise ratio, once again, if the signal to noise ratio is big, that means you are not good at hearing the speech sounds when they're embedded in noise. If the signal to noise ratio is small, that means you are doing well at hearing the speech that's embedded in noise. And so they find that these latencies are correlated with the speech and noise performance. So basically, the faster your brainstem response latency is, the better you are at performing these somewhat difficult speech and noise tasks. The quicker that response occurs and the brainstem potential, the better your ability to hear speech sounds when they're embedded in like a party or there's a lot of talk or babble going on around you. So this is connecting, once again, I guess one of the things that kind of a big picture thing about cognitive neuroscience, it's always nice when you can connect some type of brain measure to some specific behavior. Correlations of brain behavior correlations are a common, interesting aspect of cognitive neuroscience. And this is definitely an example. The brain measure here is the latency of these brainstem potential responses, and the behavioral measure is the speech in this particular figure, and these are speech and noise task performance. Okay, so a little bit more about brainstem potentials, and then I'll talk a little bit more about ERPs. I'm not for sure I'm going to get all the way to mismatch negativity today. So there's some neat stuff going on with brainstem potential studies right now. I guess here's another one. Here's another example of like the brainstem potential. This is a speech sound right here, a stimulus. And then here, presumably, is the response. This is like an idealized response, once again, of the brainstem response. And what's neat about the speech research using the brainstem. So the click research is cool, and it's really helpful for diagnosing, some various clinical conditions. But click stimuli themselves are kind of, there's not a lot going on. It's just a click sound compared to speech sounds, which speech sounds have a whole lot of information. Each sound that we produce is, sounds different from all the other sounds. So the different meanings of all our words, we're able to, tell the difference between all the different words because the words that we hear, they sound different. And you can actually look using the brainstem response of, the representation of these speech sounds in the brainstem potential. And what's cool about them is that they correlate in very linear one-to-one fashion, and to some extent, with the properties of a stimulus. So if you look at the stimulus, this is like a speech sound right here, like da. That's like the da part, and that's like the ah part right there. And then you look at the brainstem response to that. This is the latency period, like some, 2 to 5, maybe 2 to 10 milliseconds. And then you see that the signal starts to emerge right here. They call it the V wave here, then the A wave. And for a speech sound, it's more than just a click. So it's more than just the onset for a speech sound. There's kind of like the onset of the sound, but then there's kind of the microstructure of those, the thing that makes da different than do or bo or different syllables. That's what all this stuff supposedly is right here. It's kind of the different acoustic properties of the speech sounds. The fine structure is represented in this part of the brainstem potential right here, including like fluctuations in amplitude. So you can kind of see these fluctuations of the stimulus itself. These are like fluctuations in amplitude. And if you look at the fluctuations of the amplitude here, you can see that they somewhat correspond to that. So like there's the up, there's the onset of part of the speech signal right here. And you can see there's an onset here, and it goes down, and it goes up, and then it goes down for a few, and then it goes back up again right here, and then it goes down again, and then it goes up again. And there's it appears that there is a linear correspondence or one-to-one correspondence or close to one-to-one correspondence between the amplitude of the stimulus, meaning volume fluctuations of the stimulus, and amplitude fluctuations of the electrical responses in the subcortical parts of the brain. And so that's what's cool. I say here, the amplitude fluctuations in the brainstem potential correspond closely to amplitude fluctuation of the stimulus. It's like almost as if the pattern of electricity in the brain is representing the sound which is occurring out there in the world. And so, yeah, I have investigated some of this stuff, and I'm not going to talk about my research per se, but I also wanted to talk about, in some of these studies, they perform a Fourier analysis. So these are studies that are getting to some of the most complex, type of event-related potential studies, because so I guess these kind of, when you're applying the Fourier analysis, you're kind of leaving the realm of event-related potential studies. But that is something that occurs with the electrical activity right here. They'll actually perform a Fourier analysis. And once again, remember, Fourier analysis or frequency analysis is breaking down all of this electrical signal, this is the raw electrical signal, into its different sinusoidal components. So there's going to be low frequency components, and there'll be high frequency components. When you're speaking, there's a lot of different frequencies going on simultaneously. For instance, you have a fundamental frequency. Mine's pretty low, which is like kind of the carrier signal of your voice. But then the words that you're saying themselves contain different frequencies, which are usually relatively high frequencies compared to your fundamental frequency, which is usually low. The speech sounds themselves usually have a bunch of high frequency signals embedded in them. And so you can actually measure the amplitudes of those different frequencies in the speech sound. So you can actually use a Fourier analysis, and you can find, this person's fundamental frequency voice, there is around, so-and-so hertz. Or, and men tend to have low frequency fundamental frequencies, and women tend to have slightly higher fundamental frequencies. But you can get that information by performing a Fourier analysis of this signal over this kind of long span of time right here. You can apply the Fourier analysis and look at what frequencies are dominant in this. And yeah, so those frequencies of the speech sounds, I should say, are called formants. That's the term typically used in speech sounds. The different frequencies that define speech sounds are formants. For instance, da and ba, those are two different speech sounds. And the basic difference between those, like the way that your auditory system is able to hear the difference between da and ba, is that the pitches contained when you're saying those words are slightly different. The pitches in da are slightly different than the pitches in ba. And your brain, your auditory system, is able to detect those differences in pitch, and it helps you categorize one speech sound as da and the other one is ba. You're using pitch mostly. Well, that's part of one of the cues. There's other cues, I guess. Pitch is definitely one of the cues that your brain uses to help you figure out the identity of a speech sound that you're hearing. So here, these are probably the most complicated figures I'll be showing you about brainstem responses right here. But this is a brainstem response involving a Fourier analysis. So this is kind of like combining an ERP study with a frequency analysis study. So they kind of first calculated the event-related potential, the brainstem potential, and that's what this is right here. So this is the response to a speech sound. It looks like it's da again right here, which occurs at time point 0. This is the raw speech sound right here. So they say the onset occurred about right there. And then there's the vowel is the da part. That's what that is right there. And the different parts of that speech sound, da, presumably are all represented in this 200 milliseconds or so, 180 milliseconds of the brainstem response. So this is the event-related potential right here. Each of these fluctuations would be considered an event-related potential. So there's many waves or many fluctuations here. And then, but the ones that we've mostly been focusing on are those onset responses right here, like the V and the A wave. Those would be right there. But then there's also these responses that occur afterward. That's just the onset of the response, the beginning of your brainstem processing these sounds. And so the Fourier analysis, this is right here. This is just this is what the spectrogram of the speech sound. This is like the auditory fluctuations of the sound itself, the acoustic fluctuations of the sound. This though, I'm showing you these graphs can be tough, but there's actually a lot of studies that use this. Fourier analysis is a very common thing. This is a very advanced Fourier analysis. So when we talked about Fourier analysis back with the EEG studies, we were talking about frequencies that were kind of confined to 0 to 100 hertz. So like the delta band was like 0 to 4 hertz. The theta band was like 4 to 8 hertz or something. I don't have them memorized right now. But there was the gamma band, which was the highest band that we talked about in the EEG studies earlier, goes up to about 100 hertz peak. But when we're dealing with the brainstem potential, and we're dealing with the frequencies of pitches that occur out in the world, those frequencies are much higher than 0 to 100, which is the frequencies oftentimes measured in EEG studies. The frequencies here of pitches you'll hear in the spectrum, in a speech sound, might be as low in pitches, 1 or 2 hertz, or very low hertz, or they might be as high as, like, at least in this speech sound, 4,000 hertz. But so that's what the y-axis is here. This is basically all of the different frequencies. So notice also that here, they're basically dividing up each of these frequencies by itself. They're not, when we're talking about alpha, or that's like a collection of frequencies between 8 and 13 hertz. So it's like a grouping of frequencies. Here, they're not grouping the frequencies into different categories like delta, theta, and alpha. They kind of just look at the amplitude of each frequency by itself. And that's what's showing up in this graph. And these graphs, they're a little bit tough, I think, at least. There's x-axis is time, and the y-axis, once again, is the different frequencies. And then the dark colors represent amplitude fluctuations. When there's a big amplitude fluctuation, the color will appear very dark. Like, for instance, right here, this is a big amplitude fluctuation. Not shown, this might be the fundamental frequency of the speaker's voice right here. They didn't actually say in this specific paper, but you can see there's a dark band right there. I would guess that these things down here, those dark bands at the very bottom, are the speaker's fundamental frequency. All of these bands right here, this is another, this is an amplitude peak right here. So this is a frequency around 800, 900 hertz, where there's a whole lot of energy in the speech sound. Remember, these are the brain's responses right here to this speech sound right here. And what they're basically finding is that the frequencies which are really prominent in the speech sound, those formants, which are what these things are right here, there's frequencies which are really dominant in a given speech sound. Those formants, which are acoustically very dominant in the speech sound, are in the brainstem response also very dominant, which is just really neat. It's the only example I know of in the brain where the properties of something which are out in the world are represented in such a one-to-one linear fashion. Whatever the frequencies and their amplitudes are out in the world, the brain is basically representing them in the exact same way. It's taking those different frequencies. The ones which are really dominant out in the world are the frequencies which are really dominant in amplitude in the brainstem response, the Fourier analyzed brainstem response. Well, let's do a little bit more. Yeah, I don't think I'm going to get to mismatch negativity, though. I'll just talk about sensory and long latency ERPs, because this stuff is super dense, and I want to give you guys some time to consolidate. So here, we're moving on away from brainstem potentials now. So remember, those brainstem potentials were all of these things occurring really, really quickly after the onset of a stimulus. It's basically thought to be an auditory stimulus. The very first thing that occurs is that signal goes to the subcortical structures, the brainstem / midbrain areas. And that's what we were looking at, those really early, those low latency responses was the brainstem response. We're now moving later on in processing because, you know, the brainstem, subcortical stuff, that's just the first thing that happens. But then those signals keep getting sent up to the cortex. In fact, I want to show you that real quick. I think we have these figures right here. Yeah, moving up. So that's the brainstem response is supposedly all this stuff right here, but you can see there's kind of this movement upwards. The signals come in from the ears down here, then they kind of moving up, and they eventually reach primary auditory cortex, which is in the superior temporal lobe. So the temporal lobe's on the side of the brain. Superior means on the top part. Primary auditory cortex is just the first part of the cortex. The first part that processes sounds in the cortex. So it's not actually the first part of the brain that processes sounds, but it's the first part of the brain which is not subcortical. It's the first cortical region of the brain that takes input from sounds is right here. And then there's kind of an analogous thing for the visual system, which I don't have a diagram for, but the visual system, the information enters your eyes, and then the primary visual cortex is in the occipital lobe, which is back here. So for either modality, vision or hearing, sound enters, and then eventually it makes it to that first part of the cortex, either primary visual or primary auditory cortex, that first part of the cortex, which is processing the sounds. There are some subcortical stops first in both the visual and auditory system. There appears to be more of them in the auditory system. Seems to be more going on for sounds in the auditory system. But then eventually, both modalities, vision and audition, eventually reach these primary sensory areas, which are in different parts of the cortex. So let's move on here. So that's what these are right here. And you have an article, ERP Review 1, which touches on these. And so I just want to introduce some of these ideas here, and then we'll continue on with this stuff next time. So there's a slightly different naming scheme than we had presented earlier. So there's different naming schemes kind of in different fields. And we talked about the N170 earlier. That's the first ERP that we talked about. That was the face ERP. N stood for negativity. The 170 standard for the amount of time. So sometimes in these studies, the number that appears in the event-related potential wave, the number represents a latency, as in the N170 response. But sometimes the naming scheme, the numbers, doesn't represent a latency. It represents kind of the sequential order of peaks that occur in the wave. So for instance, right here, this is an idealized sensory response. I believe this is to a visual stimulus right here. And first off, I wanted you to notice the latencies that we're dealing with and all these. So now we've moved the onset of the stimulus, time point 0. And then this event-related potential peak right here occurs with a latency of about 80, 90, or 100 milliseconds, 130, maybe 150 milliseconds, you know, around 180, 200 right there. The peaks that we're talking about here, they have relatively longer latencies than what we were dealing with the brainstem response. So it's really short latencies in brainstem response. And here, this 1 does not represent a unit of time per se. It represents the fact that this is the P man stands for positivity. The 1 stands for the fact that it's the first peak right here. And it's a positivity. Please note in this graph, the negative is up and the positive is down. And that's just something you always got to check, look at the y-axis on ERP studies, because half of the time negative is up and positive is down, and the other half of the time positive up and then negative is down. So you always have to look here, positive is down. The first positive peak, P1, the first negative peak, N1. These are usually considered to be deflections of each other. If you have a big P amplitude fluctuation, you'll tend to have a big negative peak as well, because they're like almost like they're echoes of each other. One big peak here usually will result in a big peak on the other end as well. P2 stands for the second positive peak, into the second negative peak, P3 the third positive peak, and so on and so forth. So this is a kind of another naming scheme for these event-related potential studies. These sensory ERPs that we're going to be talking about, you know, a little bit now and next time, have a relatively longer latency than those brainstem responses. Yeah, so that's where I want to call it a day, actually. So let's see if I remember to, yeah, so here's the extra credit opportunity. So I think this would be a great thing to start using this. We have some time today, and I think it would be great to actually start coding with this time today your study for group project 2. As far as I understand, I talked with all the groups, and it seems like most of you guys have ideas which are doable. We kind of agreed what, you know, the strategy we're going to take for the most part, because there's a couple groups that were trying to determine whether they were able to find stimuli that would work. But either way, it's definitely a good idea to start coding your study. And I think that on my prediction, I've never had students do this before, first time ever doing something like this. I predict the coding will be a little bit difficult. I mean, I think you guys have done it before with group project 1, but I helped a lot. And so you guys will be a little bit more independent on this one. And what I want you to do is start trying it yourself. And I imagine you'll run into some problems, and you'll have some errors, or you'll have questions like, what does this do? Or, you know, this is not working the way that I want it to, and I'm having trouble, you know, changing the code so that it will work. What I really want you to do is start just getting it going. And it's not going to be a work of art right away, but basically the extra credit opportunity is wherever you are in that process. If you make an appointment with me, preferably during lab times Friday, but I can be flexible. I met with one group last weekend, Saturday, and really it's not that big of a deal. But if you make an appointment with me Friday, preferably, but sometime over the weekend, or maybe a little bit earlier, like I could probably do Thursday if you really wanted to do it early, and just talk with me about your SciToolkit code. Ask me questions. And I will basically give anyone who makes an appointment with me, and you can have everyone in your group show up at once, or, you know, if not everyone can make that, you can have like one member of the group meet me at some time and the other 3 members meet me at a different time. That is not ideal. I would much rather everyone meet at the same time. I think it'd be much productive. But basically, if you meet, you can get credit. And so the reason I've kind of suggested that potentially people within the same group could meet with me at different times is it's an extra credit opportunity, and I want everyone needs to have the opportunity to get it. And I don't want it to be that only certain people in your group have the extra credit opportunity because other people can't meet for whatever reason at a given time. So I'll be flexible with times. I'd rather meet with your entire group all at once, but if I have to kind of break it up, that's okay as well. And I will basically give you up to 3 points extra credit. So, you know, 3 points out of 0. It'll just be its own extra credit assignment. You know, 1 if you attend, you know, if you're just there, and then you can get up to 2 or 3 points extra credit depending on, you know, the quality of the questions, you know, the progress that you have made up to now in starting your study. Are there any questions about this, the extra credit opportunity? Okay, I'm not hearing anything. So I'm going to stop recording now. And if you guys want to contact, I'm going to turn off Zoom, but I'll be around. And if you need to meet back up on Zoom, I can here in a minute. Just send me an email, and I will hop right back on if you need to talk. Otherwise, I will see you next class.